Yes.
Fantastic. Okay, so I am going to actually just start a timer because knowing me, otherwise I will radically overrun and start. Okay. Right, so hey all, I'm Yo. I've just had such a delightful introduction from Pradeep. And I'm really excited to be talking to you today. So all of my slides are actually online. I'll show the link to the slides in a moment. So if you wish to actually follow along on your own, you can see those slides. The slides are also CC BY, which means that if you wish to reuse them or post screenshots or anything like that, you're very welcome to do so. And I've also put my Twitter handle on the bottom left of most of the slides. So that's at Yehudi. And I'm going to talk today a bit about open science. So again, if you want to follow along, the bit.ly URL for these slides is bit.ly. And this is a bit long, so I apologize, but I didn't want to steal anyone else's URL. So it's ISCB-SCS-open-science-2022. And maybe some kind human being can actually post that. In fact, I've got it in the chat. There we go. Hopefully you can see that as well if you'd like to follow along. And they're also deposited on Zenodo for later. So anyway, open science. So I'm going to start here with the premise that the reason that we all as human beings, as scientists, as researchers, that the reason that we do science is because we want to advance knowledge, we want to advance humanity. And if you do that science, but you don't actually share it, then you're not actually advancing very much. So our argument, the argument I'm going to be working with throughout this talk is that we have to stand on each other's shoulders and help each other and collaborate. So the simplest definition of open science, you could say, might be sharing your outputs, but I'm actually going to argue that it's the collaboration and the working with one another that actually makes it really strong and makes it most impactful, makes it most meaningful. And to tell you about that, I'm going to talk a bit about me and about some of the experiences I've had in the past. So as Pradeep very kindly put, right now I'm the Executive Director of Open Life Science, which in the name may give you the idea that I like science and that I like it open. So we try and train people through 16-week cohorts about how to actually share their work effectively. By the way, if I'm looking away from the camera, it's because I'm looking at my second screen, but I do swear I'm paying attention. And like many of you, I am also a student. I am in my final year and that thesis needs to be done by the end of the year. So wish me luck. It'll be good. It'll be fine. It'll be fine. But anyway, before that, I've been a software engineer, both within academia and outside of academia in various industry, academic spin-outs, retail, things like that. And so I'm going to talk about some of the things that I learned. As a software engineer, so I'm imagining since we're at ISCB that many of you write code. That could be maybe you write R or maybe you write Python or maybe it's something else that you like to write. My experience is primarily web-based coding. But I'm happy with a few languages. And what I would say is that bugs and oversights, they happen. It is going to happen no matter what when you're writing code. But if you have more viewpoints, then people can spot stuff. So the moment you give your code to someone else and you say, can you try this? People say, oh, that didn't work. And obviously this doesn't mean that the bigger your team gets, that like if you have 100 engineers, then you're not going to have bugs, but a few more people and a bit more varied viewpoints will help out for sure. And so I'm going to talk through some of the bugs that I've experienced. So one on the left here on the yellow, I'm talking, you may well use BLAST, you may be experienced with that. And the blog I've linked to there actually talks about what I'm going to call a skills gap or a usability bug, where the documentation in BLAST said something along the lines of returning end results. And most people interpreted that as returning the top end results, but actually only return the first end results, which could be a completely different subset of results. And so my point with this one, the reason I call it a bug is that people interpret the documentation one way and use it a different way, and it results in errors in the science. Another example, working as a scientific software engineer, is that we'd write documentation that says things like, if you want to run this on Linux or Mac, here's how you do it. Or we'd say, we've tested it on Firefox or Chrome or Safari. And actually, there's a bunch of things missing there. So we've talked about things like operating systems. We have completely missed Windows. And so a huge number of people can't partake of the science we've been doing. Or on the second one there, we don't have Internet Explorer or Edge. So there's a huge subset of people who actually can't participate. And had we had maybe a broader community, we could have tested the work that we were doing a little bit better. And another bug that is something that I've experienced when I've been writing code, or in fact, this is one that I've come across, and it's a very personal kind of tough bug or problem that you may encounter. So if you look at the pictures on the right, you can see a machine learning data set that has a bunch of different pictures of possible skin cancer to train machine learning models on. And actually, if you think about it for a minute, you may notice that this is only white people in this data set, which means that the likelihood of this machine learning algorithm actually ever detecting any cancer on someone who has darker skin is going to be much, much lower. And the example, the paper that I've linked on the bottom left is actually written by an African-American man whose wife died of melanoma. So these sorts of bugs have very real world impact if we don't try and make sure that we have a good broad team who's advising us on the gaps that we may miss. And so what I'm going to suggest is that open science can help with this. Now, I don't want to go very naively and say open science solves everything, but sometimes it can fix things. And one of the things that you can do when you have a broader community is bring in the viewpoints that might notice some of those bugs, some of those gaps that I've discussed here when you are creating your code for your research or for other people to use as part of your science. And so when I was working at the University of Cambridge on Intermine, which was a lovely open source data integration platform for genomic data, at the start, I just started as a software engineer. And then someone said, hey, yo, you use Twitter, right? And next thing I know, 3-4 years down the line, I was running all of our training well, not all of our training, but a lot of it. And I was running community calls and I was running an internship program. And so I really learned that actually people want to help out. You know, people are very excited by being involved, even if they're not scientists, but they have code skills that you can tell them how to apply it. They may want to participate. Maybe they're scientists as well, and they want to fix a bug that they found in something they've been working on. People really want to get involved when you are working on your computational scientific work, but you just need to tell them what to do. So don't assume that if you build it or if you put it online, they will come, but instead think, I have something that I think people can benefit from. How can I make a pathway so that they can work with me and I can help them work with me? And what value can I give them back? Or the way that actually a colleague, someone who works at Outreachy, put this, I borrowed this slide because I just love it to pieces. So Anna Esso, they talk about gardening a lot. They've got the most amazing houseplants that you will ever see if you ever watch their Twitter account. And they compare gardening and community management to the same thing. And they say that you know, there's so much in common. And in fact, oh, I thought I had another slide there. Okay, I was going to say, when you think about gardening, if you water it, if you take care of it, then it grows and you get a lot more back from it. You get the beautiful flowers, maybe you get the fruit. And if you don't pay a lot of attention to it, then, you know, maybe it's a cactus and it doesn't need water all that often, or maybe it sort of withers and it dies. And it basically what you put into a community tends to be much more like what you get out. So the more you do put in, the more you get back. And so I'm going to talk about some of the guidelines. You think, okay, right. So you said, let's make community for my open science project. One thing that I'll say is that it doesn't come for free. So I mentioned earlier, if I said, if you build it and they will come, maybe sometimes with really exciting projects that works, but mostly you need to put time in and then you'll get something back. You need to think, what can I do that's going to help people? So establish clear expectations. For example, let's say you've put your code on GitHub. You might want to say, here's how you contribute, please visit these issues. Or it might be that you say, here's what a non-coder can do to contribute, but make things really clear. Make it easy, tell people when you're, oops, what have I just done? There we go. Okay, sorry, my screen went blank for a moment. Make sure that you communicate with people and make it really clear how you can offer feedback if they're participating as part of your community. Encourage them and try and model the practices that you want to see. So for example, if you have your code openly, you want to use the same code guidelines that you want people to be using. You want to be nice so that people will be nice back to you, and so on. And you may say, okay, you've kind of straight away, you jumped into open science is cool and community is important, but actually it's a lot of work. You know, I'm just a postgrad who's trying to write some code to do my science. How can I get to doing this community thing? Do I even want to do the community thing? And so I'll talk a little bit about Open Life Science, which is actually the organization I run. And I'm lucky enough, I work 3 days a week, paid to work on this, but it started out as a volunteer initiative in 2019. And what we do is we run a 16-week training, which teaches people how to share their outputs effectively, to share them responsibly, because since we're all working here in computational biology, biomedical, there may well be things you shouldn't be sharing as well. And also ways to share as effectively as you can if your data is restricted, for example. And it also talks about building that community and getting people to contribute to your work, and different ways to build those pathways and get people involved and give them a value exchange for what you're doing. So if that's something that sounds of interest to you, I'll talk a little bit more about the program. Technically, applications for the program closed, I think, on Thursday, but if it sounds interesting, ping me by email and we'll see if we can sneak the applications in. But first, I'll tell you what the what the program is. I'll briefly also just mention the 4 lovely people on the side. The bottom one's me. But my co-founders and organizers, Bernice Batu, Ming Tang, and Malvika Sharan are also incredible open scientists who can actually teach you and model so much amazing open science. So I think I said this earlier, but we believe that for science to be effective, it has to be shared with others and made freely available if you really, really want to do good science. And so we try and help people, that's individuals or groups, actually learn to share open science. So people tend to work on a project with us, but we're more about teaching the skills about sharing, with the idea that you can take that to any project or anything that you do later on when you're working in research, whether it's inside or outside academia. And one thing that you may straight away say, you know, I'm worried about being criticized, you know, my work is messy, or it's not ready, or I haven't put metadata out, or maybe I'm going to get scooped. And we say, yeah, we know. People do worry about that. And that's part of the reason why we try and provide the training, is to help people learn about how to do it in safe and effective and useful ways and grow confident sharing their work without feeling vulnerable. And so we actually sort of go step by step to apply these processes to our work. And I'll walk you through the program so that you can get an idea about what that means. So we ask everyone who participates in our 16-week program to have a project. That just means something, it could be an idea or it could be an existing project that you want to work on that has some sort of open science related work. And so over the first few weeks, you meet a mentor you work with one-on-one, or your group on one to the mentor. And then every other week, roughly, you meet a cohort of people who are going through the same experience, learning about different open science techniques. So just skipping through some of the weeks, on the top left, welcome. We start talking about project visions and an introduction to working together openly. We talk about creating roadmaps so that you can share with other people where your project's going, because if someone hops on and they find a website or a GitHub repository or whatever it may be online, and they can see what you're doing next, they're going to have a better idea about whether or not they want to contribute. We talk about things like sharing hardware, sharing data, sharing software. We talk about project management. Sometimes, you know, having those practical parts of breaking down a project into smaller steps can be very helpful. And we talk about creating inclusive communities. And then we also talk about careers. So I think I mentioned earlier, you know, we don't believe that research has to be only in academia. There are many different places that researchers and research support roles can be. And we also begin to talk about a bit more advanced things like DOIs, publishing your code, preprints, ally skills, where you actually use any privileges you may have to stand up for other people. And then we talk about things like data and software management. And finally, the last week, the thing that we have is we ask people just to present what they've learned over the 4 months that they've been in the program. And this tends to be really tiery, really special. I know my time is nearly run out, so I'm going to move on. This is where we are today. The global map here, there are a huge number of countries who've participated in open life science. So you get a chance to build a really global network, and there's almost 400 people who've been through the cohorts that we've had so far. There's been 5 iterations of the program. And these are all of our mentors and our experts and the participants in the program. And so I'll just, oh, that's my alarm saying I have to stop, dismiss. Okay, I will wrap up by saying consciously plan what you're doing when you're doing open work, because that's the most effective. Tends to correlate with market performance, but I'm going to skip over that slide and just give it back over to you, Pradeep. Thank you for having me.
Amazing, you. I think we should have planned for 1 hour talk by you, and 15 minutes is too slow. Thank you very much. The audience, please put your questions in the chat. I think just take a break and then also maybe think about and drop the questions. Meanwhile, let's invite our second speaker, Dr. Ming Tang, and who is popularly known as Tommy Tang. Welcome. And so Tommy is now the Director of Computational Biology at Immunitas Therapeutics. At Immunitas, they employ single-cell sequencing platform to dissect the biology of immune cells in human tumors. A very important topic to deal currently. Tommy describes himself as a computational biologist working on genomics, epigenomics, and transcriptomics. And Tommy uses R primarily for data wrangling and visualization in the tidyverse ecosystem. Tommy uses Python for writing snake-make workflows, which is also very important for us while working on computational and bioinformatics projects and reformatting data. And Tommy is a Unix geek learning shell scripts almost every month. And that stresses the point that the learning never stops and we should keep on learning. And he also cares about reproducible research and open science, and which we will be hearing now. And thank you, Tommy, for accepting the invitation. And now the floor is yours.
Thank you very much for your nice invitation and also introduction. So thanks, Yo, for educating us on how to build the open science community. Okay, so it's my honor to present at the ISMB Student Council Symposium. So as it was introduced, my name is Ming Tang, or you can call me Tommy. So I'm currently the Director of Computational Biology at Immunitas, a single-cell genomics company committed to develop novel therapeutics for cancer patients. So before that, I was leading a bioinformatics team at Dana-Farber Cancer Institute for an NIH-funded consortium project called Cancer Immunological Data Commons. So I was trained actually in a wet lab during my PhD at the University of Florida and started to teach myself Linux, R, and Python when my advisor asked me to analyze public chip sequencing data back in 2012, and I couldn't open it with Excel. So then I did a 3-year computational biology postdoc with Rova Hack, who was leading the TCGA glioblastoma project. And after my postdoc, I moved to Harvard FAS Informatics to work on single-cell RNA and single-cell ATAC sequencing data. So today I'm going to talk about my favorite topic, reproducible genomic data science. Again, this is my Twitter and this is my blog, if you want to follow me. So I consider myself as a genomic data scientist if there's such a term. So as I mentioned, I have extensive experience in analyzing different sequencing data, including genomics, epigenomics, and transcriptomics. So I frequently share my experience of sequencing data analysis on my GitHub repositories, and you can go there and read them. So as to today's topic, reproducible research is always dear and near to my heart. So now let's dive into today's topic. So we all know how science should be done. So first we make an observation for a hypothesis, and then we perform the experiment, analyze the data, and then report our findings. And lastly, we invite others to reproduce the results. However, we often hear that one cannot replicate the published study. So there is a reproducibility crisis going on here. So you may think experimental studies may be hard to reproduce because the experiments were performed under different environments, different people handled the mice, or different reagents were used. One may think computational studies should be easier to reproduce. However, according to Titus Brown, a bioinformatics professor at UC Davis, 95% of the studies fail to include details necessary for replication. So it is really hard to build off of research like this. So let me give you an example. So there was a scandal in Science that a researcher at Duke faked the data and built the results on a flawed analysis. And Dr. Keith Beckley from MD Anderson downloaded the microarray data and could not reproduce the results claimed in the paper. So through a detective investigation, he finally found the original authors had one row shifted in Excel and got all the gene names wrong. So unfortunately, the clinical trial built upon the results has to be stopped. So this speaks to the fact that how important we should make our analysis reproducible, both for your own sake and the patient's sake. So I highly recommend you watch this video, and he's a really fun person. So here's another example in this paper published in Science titled Rearrangements Burst Generate Canonical Gene Fusions in Bone and Soft Tissue Tumors. And in the method section, it says we detect gene fusions in regions of genomic complexity using an approach that integrates multiple independent fusion algorithms. And that's it. So without further details, one cannot really reproduce the results. And even how do we really trust their results? So why reproducibility is so hard? So I summarized 5 different points. So first, no raw data are available. And second, scripts or data available upon a reasonable request. And you will see this in published papers, which is, in my opinion, unacceptable. Third, a lack of method description, just like the previous example that I gave in that Science paper. Fourth, the version of the tools are different, maybe different version for R, Python, and bioinformatics tools, they all have different versions, and maybe different versions give you slightly different results. And lastly, it could be due to your machine, for example, Unix machine versus a Windows machine. So if it's so hard, should we care about it, right? Keep this in mind. You are going to do the same analysis for sure in the future yourself. This is for your own benefit. And now I am in a biotech company. Our mission is to develop life-saving drugs for patients. I certainly want my analysis to be reproducible. Then how to really ensure reproducibility? I'm going to give you a high level of the tools that I'm aware. First, Git version control your code. And second, use the Jupyter or R notebook to document all your analysis. Third, to use Docker or Singularity to containerize your computing environment so others can reproduce your analysis using exactly the same environment. And lastly, unit tests and continuous integration of development, or CI/CD. Those are more like more for a software engineer. So I'm sure we all experience this on versioning our Word documents. So we keep adding the version number after final _red 1, 2, 3, 4. And the same thing for code. So we should use Git to version control our code. So Git is a software for tracking changes in any set of files, usually used for coordinating work among programmers. And Git records all the history of your files so you can easily revert back to any previous version. So GitHub and GitLab are websites that host your Git repositories. So I highly recommend putting your code for published papers on GitHub or GitLab so others can replicate your analysis. Notebooks. So a wet lab member has a notebook to document his or her daily experiments. So what, when, and how he performed the experiments. And similarly, a computational biologist should have a digital notebook to document all the analysis. And Jupyter Notebook is Python flavored notebook, although it supports other kernels such as R. So the nice thing about the notebook is that you can put the documentations together with the code, and the code is evaluated in inline. So you have the documentation, your code, and the results of the code in the same document. So I use R often, and I love R Markdown or R Notebook. So it is similar with Jupyter Notebook, but it is R focused. Similarly, you can have this code in your document and the results of the code, and also the figure generated by the code, which is very nice. So in addition to document your analysis in the notebook, you should always automate the analysis as much as possible. I really like this cartoon. So on the x-axis is the task size, and on the y-axis is the time spent. And there are two ways to approach the same problem, the geek way and the non-geek way. So the non-geek way does it manually all the time, and the geek way does it manually first, but then gets annoyed, so spend time writing scripts to automate. And once one has the script, he can run scripts, and when the task size increases, he beats the non-geek way in the long run. And there's a really good side effect of automation, because the best documentation is automation. If you automated the analysis, the documentation is self-contained. So make sure you write scripts for everything unless it's not possible. For example, you have to do some manual editing, but you have to document all those steps. So important things I say 3 times. And internally, actually, we use mkdocs to write documentations. Mkdoc can translate a bunch of plain Markdown files to a nice website. There's some tips for automation. So if you have a repetitive simple task, put them into a single shell script, myroutine.sh. So instead of typing single commands one by one, you can just type one command, myroutine.sh. So you can also use a good GNU make. So once it was designed for compiling software, but people use it for data analysis too, especially when your analysis has multiple steps and each step using different tools. And make is a nice tool to, I consider it as a glue to glue different steps together. And more recent workflow languages such as SnakeMake, a Python extension of Make, and Nextflow, Widow are your friends too. So in this repository, it collects hundreds of tools that can be used to write pipelines. And now let's talk about the containers. So you might have used a virtual box, but think of it as a virtual machine that encapsulates all the software libraries and configuration files. And then you can ship this virtual computer to your friend, and he can use exactly the same computing environment to do the analysis. And this in part solves the reproducibility issue. And I put 2 links here, and if you want to learn more about Singularity and Docker, you can go there. And the number 0 problem for bioinformatics is converting formats of input and output between different tools. And the number 1 problem is then installing the tools. Sometimes it takes me like the whole morning just to install a tool. It's very difficult. And there are so many different bioinformatics tools, and many of them have different dependencies on other software. For example, a particular tool is Python 2, and another tool is Python 3. How can you do the analysis if it is dependent on both tools? So Conda is a package management tool for any language. One can set up 2 different Conda environments, one for Python 2, the other one for Python 3, and that solves the problem. And BioConda is a life science-centered channel in Conda. So you use Conda or BioConda to manage your computing environment to help with the reproducibility. And finally, project or organization. This is really underrated, and not many schools teach how to organize a computational biology project. I recommend you to read this paper, A Quick Guide to Organizing Computational Biology Projects. And I follow a similar folder structure. So on the top level is the name of the project. Then you have the different subfolders, for example, the documentation folder, the data folder. The source file folder contains scripts, and the bin folder contains the executables. and the results folder, and you prefix the results or data with the date. So having a fixed project structure can help you to produce your results in the future. And finally, I want to recommend these two papers: Good Enough Practice in Scientific Computing and Best Practice for Scientific Computing. So both are written by the software carpentry communities. So if you want to learn more about what should you do, what's the best practice or good enough practice, go and read those two papers. Definitely will help you. Now one can take one step further. So Workflow R is an R package to translate your analysis R Markdown files into a web page. And one can easily browse through the code and the documentation and the figures on a website. Pretty neat. So I want to end this presentation with this blog post that I wrote very early, actually, during my career in computation. So first, you need to realize computers make mistakes. They can give you nonsense results and exit without any error. So make sure you do extensive tests. And also for the analysis that does not have a ground truth, please do some sanity check. Talk to the biologist, whether it makes sense biologically. The results, genes that are upregulated that identify in your analysis, does that make sense in the system or not? Second, share your code. So share your code first. People can find errors in your code, right? Even if your code's right, people can find ways to improve your code, and you learn from them. Third, make your code reusable. Don't hard code, for example, your scripts. If a file path is being used as an input, make it as an argument. Fourth, modularize your scripts and use, if your analysis have different steps, write different functions or modules for different steps, so you can easily recycle and reuse them. Heavily comment your scripts, because it's going to benefit the future yourself. You're going to read your own code sometime in the future. And lastly, you need to make your analysis reproducible. Okay, I will end up with this last slide, acknowledgement. I want to thank Samir Amin in Vakar Lab, where I did my postdoc, and he taught me a lot of things. And I also want to thank Titus Brown, Data Carpentry, and all other people who share their wisdom on the web. Thank you very much.
Thanks a lot, Toby, for also sharing your wisdom on the Twitter. It's really helpful for all of us.
Let me stop sharing. Thanks. Am I still sharing it?
Yes.
Okay, let me see. How do I unshare? Okay. Stop share. Okay.
Yo, are you there? So let's have a questions. Okay. I put up. Okay. So let's. Are there any questions in the Q&A area? Let me check now. Okay. So I'll start with first. Hi, Olatin, please ask your question.
Okay, thank you. So my question is to the second speaker. That's Ming Tang. And that was a great talk, by the way. Thanks. I just loved everything. So my actual question is, you showed a slide that talked about how to arrange life science projects, like the data, you have the bin, the source, and some other directories. But I was wondering where you had docs. I was wondering where you would put notebook for Jupyter, related stuff, because now that falls in the category of documentation and code and results, like if you're doing our Markdown, or I was just confused. I followed everything except that particular slide.
No, I think as long as you have your own structure, that's fine. This is just an example. For example, I put all the R Markdown files in the scripts.
Okay.
Of course, all the data that will be in the data folder, and I will make it untouchable. So you do chmod u minus w, so you cannot write the data. And I always take the data in the data folder, use the scripts in the scripts data folder, and generate results in the results folder. So in this way, you can also reproduce your analysis. Yeah.
Okay, so you wouldn't recommend me just creating a new directory called notebooks for the notebooks part that falls into like 3 categories overlapping in your structure?
Yeah, I would say as long as you have your own consistent system and you stick with it, that's good.
Okay.
Yeah, you don't have to actually to follow that example, but particularly, yeah.
Okay, cool. Thank you. I would probably like to, yeah, I'll follow up with you. I think I have your hand over. Thanks.
Sure.
Yeah.
Thanks, Olaitan. Now, Tommy, we have a similar related question where Laiba asked, is Conda compatible with Python only or with R as well?
Conda actually is a package management tool for any language. So all the different tools are Python, even Java, they're all there. So you can use it. Yeah.
Thank you. So next, any more questions? No. So maybe I'll ask one of my questions. So to first Yo, what motivates scientists to follow more open science principle? Are these motivations currently we have sufficient for the growth of open science?
Classic question. Thank you. So I'm going to say we're getting better. I don't know if we're good enough yet, but I think with the motivations that we have around us are getting better. So some funders require things to be open. For example, it may be a condition of your grant, depending on what funder you have, that you deposit your code openly, or that you deposit, or it might be that a journal says you're supposed to deposit your code or your data, or, you know, different open access options. But I think that on an institution level, it's not sufficiently encouraged, and that perhaps we could do a better job of expecting people to write open science good practice into their grants and dedicate time to it. Because I think that even though people acknowledge the importance, they often feel like they don't have enough time to put towards it. Especially things, I was reading a paper about intentions versus what people actually did around sharing their data. And a lot of people know that it's important, but also think it's going to take them so long to prepare their metadata that they don't share things because they're worried that their data may be misinterpreted or misused. So we need to be able to, you know, say to our bosses, to our funders, to our institutions, I'm doing things that aren't writing papers. I'm doing best practice for my code and for my data as well. I'm sorry, Pradeep, you're on mute.
Thank you. So how should we encourage this kind of culture for the students? Like most of the points you mentioned was towards the researcher who are at a stage of asking funding. So, but most of our audience are like on a student level, which is PhD, masters. So,
great point. And that's kind of nice. I would honestly say, unless your PI is watching you every minute, they're going to assume that you're trying to do the best job that you know how to do. And as part of that, that you should be modeling the culture that you want to see in the future. And teaching your peers about it as well. So if there's a practice that you know about, let's say, so I really enjoyed Tommy's talk. I was just like, I wanted to +1 everything you were saying, put your code on GitHub or GitLab, right? All those things. But if you know how to do it and someone else doesn't, help them. Make it enjoyable, make them not want to bash the computer and say, I hate the command line. You know, just share that culture as freely as you can, because then they'll help you at some point, otherwise later.
thanks, you. I would like to ask the question to Tommy now. From we collected some of the responses and questions beforehand. So one of the respondents says the main issue with reproducibility is it is not well established the sharing of data for allowing easy exploration of published data. Example, because not all metadata available authors are not willing to share their work or analysis, and journals not verifying the quality of published data also the repositories, GitHub repositories they share on the different level of robustness data. So my question is, so how could we encourage our young audience right now so to not to follow these bad examples from the so-called experienced researchers, but develop the reproducible skills and the lifestyle of reproducibility early in their careers?
Right. Again, lacking of a standard, I mean, that's probably a problem. So we don't have a standard to follow. For example, most of data, genomic data, like we upload to Geo, for example. But even for Geo, like you will find the metadata is a kind of a mess if you want to actually do some meta-analysis across different data sets. For example, single cell RNA sequence data, like different data sets, they have different levels of metadata, and people may just don't even put the necessary metadata there. Like, for example, when the data were processed, or when the experiments were done, like maybe they were done in different batches, different time point. Then you could have batch effect just purely based on the processing time of the tissue or things like that. So I would encourage, if you have your paper published, make sure to include as much information as possible. I know it will take your time, but definitely will help people in the future to reproduce to reuse your data, because the value of your data is not just the paper itself. In the future, it can help other people as well. So make sure you have as much information as you have here. Yeah.
Thanks, Tommy. Yo, please.
Thanks. Tommy, I enjoyed your answer there. But I was just thinking of another thing that maybe students and postgrads and postdocs can be doing is if you are reviewing a paper, check these things. So an example, I remember a year or two ago, I was reviewing a paper and it was actually about reproducibility in biology. And their data was on Google Drive. I'm like, okay, it's great that you shared this, but what if someone leaves the institute or moves that file? That data is going to disappear. So please put this on an institutional repository. I mean, a lot of people say, no one has time to review these things, but I would argue that if we're computational scientists, that we should be reviewing the methods, and code is our methods, right?
Yeah, thanks, Yo. That's an excellent point. So if you're a reviewer, make sure you check that. I think that's a really good point. For example, if the paper is about the software, a piece of software, for example, like you need to make sure the code is on GitHub somewhere, and you need to really try actually install it, whether you can easily install it or not. So that's usually what I review bioinformatics paper. I will just go and try to install it myself. If it doesn't work, then I will just tell them, okay, make improvements.
Maybe a quick follow-up question on this note. You both have been talking around the people who do reviewing and then like testing the softwares or the data what they claim in the paper, right? So, but is the current incentive system of like this is a lot of hard work for the reviewer, okay, at least let's say in the people within in their early career stages. So is the current incentive system is sufficient, or what kind of incentives do we need to like cope up with these efforts the people put other than their motive of encouraging open science and reproducibility?
I'm willing to take this one first. So I don't know if this is radical, but I think we should pay reviewers. It's a lot of work. Every academic I know, the first paper they're asked to review, they're excited, and the second one, and the third one, and the 19th one, they're like, oh God, I don't have time for this. And when, as academics, as researchers, as people who support research, we always have more than we can do in our time. There's always going to be volunteer opportunities. And we don't equally have time, right? Some of us have more time, some of us have less. We might have caring duties, we might have families we want to spend time with. And if people pay you for the work you do, you can choose much more easily what you want to do, and you're more likely to want to, hopefully, do a decent job.
Thanks, Yo. So we have questions in the Q&A, so I will take one question. So where Melike asked regarding the metadata, it's great to encourage people to submit as much metadata as possible. But do you know any initiatives that actually encourage institutes or journals and databases to enforce the submission of better metadata?
No, I personally don't know. Yeah, so I think Geo is doing a decent job. Yeah, but we can do better. So yeah, but I don't know an organization that is enforce us or us to do a better job on metadata. Yeah.
So how about? Oh, sorry. Go ahead.
Sorry. I was going to say, it's such a balancing act, right? Because if you say have lots of metadata, you have everything good quality, but people won't be bothered to share. They're like, oh, it's too much work to upload this. And it's like walking a tightrope, trying to not too much to one side or too much to the other side.
So what do you think about the report? I mean, the efforts behind Array Express and Expression Atlas by EMBL EBI. So I think they try to enforce submissions at Array Express level, and then they also they have curators at their institute, and then do Expression Atlas. So do you think such kind of efforts are more and more needed?
EMBL probably is doing a better job in that sense, according to what you just said. I think definitely it helps, because if the data repository, the owner of the data repository makes a strict actually policy, and if you want to publish your paper, you have to actually record more detailed metadata. And I think that actually will be beneficial for science in the long run, for sure. Yeah.
Thanks, Tommy. So we have another question from Ina Maria. So what is your thought on whether or not institution, journals, or grants demand researchers to follow the reproducibility principles? How much time will still pass until this may happen? Should it be required or remain a free choice? If demanded, who could control and decide the degree of reproducibility is still acceptable? Would that create a new kind of profession?
You want to answer this question?
Okay, I'll give it a stab. So I'm just looking at this question. I think that people will want to make it a carrot, right? They'll want to make it something that you look good if you do it, and that if you don't do it, it's not going to be the end of the world. And I think that's just because of the time pressures around this, but more importantly, because of the standards. So if we had so much good information about reproducibility, primarily in R from Tommy's talk, but then there's Python. I like to write JavaScript. I know that's almost heinous to suggest, but I love JavaScript. And people write so many different types of code, so many different software domains, research domains, that I don't think it's practical or realistic to have a clear defined set of definitions for what we need for each set of each paper. There's no way that we could ever cover that. And so I think that we should perhaps as reviewers just recognize, I would rather do 3 reviews in a year well than do 15 reviews really fast. A paper I worked on, someone said, I haven't tried this app, but I think it sounds great. That was literally on the review for the application that we published. It doesn't make sense to me that we do that. We should just, as reviewers, as people who are responsible for science and peer reviewing it, I think we should take it on ourselves to be the ones who are auditing the quality.
I think for the reproducibility thing, some funding agents at least require you release the data. It's really unfortunate. Many of the papers, they even don't release the data, and that's unacceptable. And at least many of the funding agencies, they require you, to release the data once it's published. So, but in order to actually ensure reproducibility, that's a much harder, a much higher bar actually to reach, I would say. Yeah, but that's actually in the long run, it beneficial. It benefits all the research, because you think that maybe 80% of the researchers, they are not reproducible, and how do you really trust the results? I mean, all the researches are built on the previous research, right? So you want to make sure your research is solid, reproducible. Yeah, and especially when I'm in a biotech company, we are finding drugs for the patients, and you can't do something, sloppy. That's my answer. Yeah.
Thank you, Tommy. I'll quickly shift from the discussion what we are talking. So one of the response we got was, my view on open science is the accessibility of data, code, resources to everyone who do science, and a platform where everyone can help each other and integrate their vision, work, and collaborate with each other. So, the question is to you. So do you think anything is missing from this list? Is it only the accessibility of data, code, or resources, or are there any open science principles are missing from this list?
Lovely question. I think in terms of some of the ingredients that you need to build that open science recipe, there's some really good basics there. I mean, data, code, if you don't have those, then your science can't be reproduced, which Tommy has pointed out so nicely is pretty important, But I would say that there are a few other things. So I'm not a wet lab scientist myself. I accidentally ended up in biology after coming from a computer background. So sometimes I have to ask the people who know more about biology than me to explain things in very gentle terms. But I know that there are places where you might have open hardware, for example. A PCR machine, if you're buying a big commercial one, is ridiculously expensive. And I know that there are efforts to make open ones that people can make themselves in relatively low resource environments. And that means that you can maintain it more easily. And there's many other aspects. For example, I'm veering into the area where I don't really quite understand the science, but are your reagents open if you're doing lab science? What other tooling do you have that you could share? And what's it made from? Is it made from things that are affordable and easy to access? If you give me a list that requires uranium for your open hardware, I ain't going to be able to build it. So I mean, you can sort of think how far down do the turtles go? So there's a lot of other things that you can do. I will also argue that community makes it better, because I mean, I guess that's what my talk was. I guess, but that also involves thinking about the resources that your community has. So I have reliable power 24/7, and internet, it's on most of the time, but some people don't. People who participate in open life science sometimes call in from their phone because they don't have power. There's rolling blackouts, for example. And I would argue that having those things is an important part of open science, or understanding and making sure that you can compensate for it if not. And so thinking about the equity of participation is an important part of building an open community. I'd argue, don't say if it's open, anyone can contribute. Say instead, it's open and we want to help people, as many people as we can, to contribute.
Tommy, would you like to add?
Yeah, I actually really appreciate this question. So open science not only about have access to the code, to the scripts, but access to a community, and which Yehudi is building, which is very nice. And people can come together, teach each other, help each other, come up with a common standard together. I think I mean, once you have a good community, and that everything else will become much easier. But again, it takes gardening, takes a lot of work. Yeah.
Yeah, I like the community. When you say the community, like the Open Life Science community is doing an amazing job. But I would also like to add on that, isn't it that this program should not be outside our own curriculum? Like it should have been fortunate, like has been in the our study program, like for example masters or postdoc. It should not be we should make these resources available to people as easily as possible, not help them to go out of their normal work life and then spend their effort, isn't it?
Honestly, I think one of the biggest failures is that I'm running an organization outside of academia for the hopes that I can change academia. And I think it's really, really unfortunately sad that that is the case. And that, like the carpentries, it's non-academic, right? I mean, it's for academics, but it's filling a niche that isn't filled in universities sufficiently. And I think if you look at a lot of open science and code-related training, you find that actually these don't fit very well in the system, and people have to go outside the system to do it. But I hope that every one of you who's here listening today can think, when I'm running my lab, I will try and make sure that my students can do this, and that they won't be afraid to say to their PI, can I share this, without and worrying about getting in trouble or spending too much time on it, and that we can change that culture together.
Thanks, you. So I think I must ask this question, which is the one of the least spoken questions, like the one of the difficulties in actually open science, and also which affects the reproducibility, is the reach to the remote areas. And because of these profit focused organizations or journals, the knowledge is only as a privilege for some people, not everyone. So how should we overcome these difficulties, and what should be the focus of the academic community, or the NGO communities, I would say, like non-profit communities who are trying for this open science and reproducibility?
Tommy, I wonder if you have thoughts. Yeah.
Prandeep, can you just reframe the question a bit? Yeah.
Okay. I mean, it is still accessibility of papers, and the knowledge we have in the community or the research is still a privilege. Right. Not everyone can access the data or the applications. Right. So what should we do to make it reachable to everybody?
Yeah, that's a very good question. So we all know the publishers, they are very greedy. You pay a lot for publication, and they charge you for reading them, right? So that's why initiatives such as like bioarchives, I think those are good alternatives, and people can put your paper on bioarchive. So even you can put your paper on bioarchive and have that version, and also you can publish later, and you can even update the version, constantly on bioarchive. So if other people do not have, for example, subscription for Nature Science, and then they can still get a copy, from bioarchive. Yeah, so I would like to recommend people to deposit their papers on there. And of course, it's also for your own benefit, because it's a faster way to disseminate your science. So it's much faster, because you can take years just for review and revision.
Before you comment on it, I would like to add one point. So there is a possibility of for us, even though we don't deposit on bioarchive, which we should be doing, but still some of the journals allow different levels of sharing the papers. For example, the published version, or the accepted version, or pre-journal formatted version. So what are your thoughts on this kind of thing? Like, how should we encourage this?
It's difficult. I don't think I'm ever going to blame a researcher for not being able to afford an APC, right? I would say maybe consider if you can go to a journal that will at least allow you to preprint. I really did like Tommy's suggestion, just make sure that you can share it somewhere else, if it's green open access or something like that. And ultimately, things like open access have the other side where you end up, rather than with people who can't afford to access the science, with people who can't afford to publish. And even though there are things like fee waivers, we all know that they don't cover enough scenarios, and that people are looking around under their couch cushions trying to find extra pennies so that they can afford to publish when it might be more than a month's or a year's wage. It's hard because we live in capitalism, and unfortunately we have to be practical about the fact that that is how our system is. But I'm just going to continue with not making people feel bad for existing within the constraints that they have, whilst doing your best to practically encourage the things that they can do. Is probably my honest answer here.
Okay, thanks, Yo and Tommy. We have one more question. I mean, the time is up what we planned. So if you're still okay, I'll ask the last question we have here. Thank you. You have been so kind. So Camilo asked, what are your thoughts about published applications or methods that can be replicated with the example data of the authors, but without the required documentation to allow testing with outside data?
Question for me or for you?
Go ahead, Tommy.
No. I think actually providing an example data set actually is a very good initiative, I would say. So if you have a piece of software, make sure you have an example data. But of course, you have to document your software in terms of what's the input look like, what's the output look like, what's the requirement of the input, right? What are the rows, what are the columns? I think being explicit for those requirements definitely will help. So yeah, what's your thought here?
I've got to be honest, it makes me a little bit cross that people would do this. A while back, I worked for a little while at a philanthropic funder, and one thing I wrote briefly was grant conditions for people who are writing software, and it included that you have to have documentation. Of course, and how much documentation is good enough. It's very squishy, but it's like, to my mind, cooking with a recipe versus cooking without a recipe, right? If someone gives me spaghetti and they give me sauce, I can probably make something that will taste the same. But if you give me a bunch of components I've never used, I don't know, rice and fish and seaweed, I'm not going to make sushi. If I've never used that code, let's say you've given me Julia. I've never run a line of Julia in my life. You give me some Julia, no documentation, I cannot guess how to run it. You give me JavaScript, I can guess. So I guess my point is sometimes people can figure it out anyway, and other times it's going to end up messy and awful. Does the recipe analogy make sense there? Give people the recipe, it's better.
Yeah, I think I like that analogy. Internally, like in my group, what I do is that, okay, if one of the group members write a piece of software or a workflow, document it, and I will ask another team member just to follow the documentation to see if the other people can do the same analysis or not. If he can, that means okay, other people can do the same thing. So that's usually what I do.
Yeah, that is perfect.